Hello friends, welcome back to the channel. Today we are learning biotechnology. To put it very simply, biotechnology means using living organisms for our benefit, for the human welfare. See, as we already know, bio means living and technology is the scientific application of the knowledge on living organisms for extracting certain products from those organisms. Biotechnology is not entirely a new concept. We have been using biotechnology since time immemorial. For example, in the preparation of curd from milk or in the preparation of alcoholic beverages from Saccharomyces cerevisiae, which is unicellular fungi, that is yeast. Baking industry, in preparation of vaccines, in the preparation of antibiotics and many other areas, for example, gene therapy, and many more, which we are going to learn in this chapter. The European Federation of Biotechnology, abbreviated as EFB, gives a more scientific definition for biotechnology. Take a look at it. The European Federation of Biotechnology defines biotechnology as integration of natural science and organisms, cells, parts thereof and molecular analogues for products and services. What does it mean? Now, we integrate natural science, that is our scientific knowledge, on entire living organism or a simple single cell of the organism or parts of the cell. Parts thereof means not entire organism, using part of the cell, an organelle or just an enzyme, etc. And molecular analog for products and services. Now, what do you mean by molecular analog? A molecular analog is a molecule which closely resembles the original natural molecule produced by the cell or the organism. Putting all this together, we use natural science, that is the knowledge that we already have on the organism, or we use a cell of the organism, or we use part of the cell. It may be an organelle, it may be a enzyme, or it may be anything which belongs to the organism. For the products and services, what may be the products? The products may be antibiotic, it may be a vaccine, it may be a service. Biotechnology has wide applications. Even the in vitro fertilization that we perform for treating infertility is also biotechnology. The earlier examples that I quoted, like preparation of curd, preparation of alcoholic beverages, or preparation of vaccines, etc., are very basic uses of biotechnology or the basic applications of biotechnology. In fact, modern biotechnology uses recombinant DNA, which is abbreviated as rDNA. What is rDNA? rDNA is prepared or synthesized by joining the DNA from two different sources. Why do we have to join DNA from two different sources? Again, for the product. DNA has many genes. Genes express by producing proteins. That protein is our product of interest. That product is exactly what we need, right? Product and service. We will try to understand our DNA as the chapter progresses forward. For better understanding of the concept, it is good to get familiar with some terminologies. The first terminology which has to be understood is clone. Clones are resultants of asexual reproduction, or offsprings of asexual reproduction are called clones. Why is it so? When amoeba divides through binary fission, two daughter amoebae are produced, which are 100% identical to the parental cell. It is because in asexual reproduction, only a single parent is involved. So there is no new genetic combination available. Only the genes from a single parent are passed on to the daughter amoebae. Hence, they are identical to the parent. Clone can be defined as identical copy of cell, organism or DNA molecule. Another terminology to understand is vector. Yes, the word vector, which we had already come across in human health and diseases. If you remember, female Anopheles mosquito would act as vector for transporting malarial parasite into the human body. Here also, vector is a molecular vehicle which carries or transports DNA molecule. Commonly used vectors include plasmid. See, a plasmid is an extra chromosomal circular double stranded self replicating DNA which is present exclusively in prokaryotic organisms, that is, bacterial cells. In addition to the chromosomal DNA, extra chromosomal circular double stranded DNA is present in prokaryotes. This autonomously replicating or self replicating DNA is known as plasmid. This plasmid is very useful in the transfer of DNA from one organism to another organism. Let us try to understand how a plasmid acts like a vector. Look at this diagram. We have a cell which contains a gene which is useful to us. This gene is called gene of interest. The gene of interest is extracted from the whole genome. It is taken out, and similarly plasmid is also cut out using enzymes called restriction enzymes. The source DNA and the plasmid DNA are cut open using the same restriction enzyme. Now, these two are joined to form recombinant DNA. How can we join gene of interest with plasmid? We can use ligase. The same ligase enzyme which was used in joining the Okazaki fragments which were formed during replication of DNA. The recombinant DNA has DNA molecules from two different sources which are represented using two different colors. Our DNA is capable of transferring the gene of interest into another organism. This is how a plasmid can transport DNA from one organism into another organism. Hence, plasmid acts as vector. Vector is a molecular vehicle. In this whole process, we are altering the genetic makeup of this individual. We are adding this gene of interest into this host organism. By adding this extra gene into host, we are changing its genotype. When genotype of an individual is changed, eventually it leads to change in phenotype. Altering the chemistry of genetic material, that is DNA or RNA, is one of the principles of biotechnology. Biotechnology is based on two core principles. One principles of BT is genetic engineering. Genetic engineering simply means to change the genetic makeup of the individual or to alter the chemistry of DNA or RNA, which we just did. How did we do it? By adding a gene of interest into the plasmid, we changed its genetic makeup, right? And this recombinant DNA was given to the host cell, was introduced into the host cell. By adding it into host cell, we changed the genetic makeup or genotype of the host. Due to change in genotype, the phenotype also got changed. I want you to answer a fundamental question. Why are we doing all this? What is the reason? What is the purpose of doing all this? What is the purpose of changing the genetic material, genotype, phenotype? Why all this? It's because This gene of interest is capable of producing a product, that is a protein. That product is actually what we want. We are already familiar with the central dogma of molecular biology. What does it state? It states that genetic information always flows from DNA to mRNA, from mRNA to proteins. Right? We have gene of interest here. Gene is nothing but the DNA. The DNA will form mRNA. The mRNA is translated into protein. This protein is the useful product that we are getting from doing all this process.